day everyone, I'm Mary Ann Montecalvo, Bised 2B at Central Bicol State University of Agriculture. And for today's video, I'm gonna show you the simulation of different stages of mitosis and meiosis. But before that, let's define what is mitosis and meiosis first. Meiosis, also called reduction division. Division of a germ cell involving two fission of nucleus and giving rise to four gametes, or sex cells, each possessing half the number of chromosomes of original cells. In mitosis, a process of cell duplication or reproduction during which one cell gives rise to two genetically identical daughter cells. Strictly applied, the term mitosis is used to describe the duplication and distribution of chromosomes, the structure that carry the genetic information. Now, in the left, we're gonna have the diploid cells with 46 pieces of DNA. That's gonna go through on mitosis to make 46 pieces of DNA. The cells start and end with diploid. On the right side, these cells will start with 46 pieces of DNA diploid. And they go through meiosis to make two cells. But there's the second round of meiosis. There's two cells divided again into four. In each of one, have half amount of DNA. So on the left, we're gonna have a cell that undergo of mitosis. And on the right, we're gonna have a cell that undergo of meiosis. So before we get started, I just wanna make a count. There are four strands of DNA called chromatids, lost linear strands of DNA. And these cells on the left and cells in the right as well is gonna have four strands of DNA or chromatids to start. Now we're gonna compare that until the end. This key right here is gonna help us visualize where the DNA came from. Where the chromatins and chromatids, the red threads are DNA that inherited from the maternal side of family, from your mother, and black threads are DNA chromosomes inherited from paternal, from father sides of family. So here we have the cell on the left is the interphase as a stage of interphase. So this is prior to mitosis. Now what happened here is chromatin is duplicated to the four strands to become 8. And on the right side, the same thing. The chromatin is duplicated. So far, the cell really can tell you the difference between the two processes. As we move on into prophase of mitosis, on the left, the nucleus will dissolve well in prophase number 1 of meiosis. On the right, the same thing, the nucleus dissolve. So far, this really notice distinction between the two. And now let's move on prophase of mitosis. On the left, we're gonna see the loose linear version of DNA called chromatids coil. In this X-shaped, structured called chromosome, and on the right hand side, the same thing. Notice how there are two chromosomes number. One in red you inherited from your mother, and one in black you inherited from your father. Same thing, these two chromosomes number 2. In human, there's 46 chromosomes, so there's two chromosomes 12 and two chromosomes 21 and two chromosomes 19. So for simplicity, I just draw a cell with 4 total of chromosomes. And as we go on into prophase 1 of meiosis, what happened next is synapsis. And what this is another two chromosomes number 1 are pulled next one another by spindle fiber, and two chromosomes number 2 is pulled next one another. Tetrad are retained from the homologous chromosomes, so we have two chromosomes 12 and two chromosomes 19 and two chromosomes 2. So that is what tetrads are. So why does synapsis happen? Synapsis set up what called crossing over, recombining the homologous chromosomes. And what happened at chromosomes? Portion of chromosomes 2 red are actually break off and reattach with portion of 2 and black. this recombined portion of homologous chromosomes of one another. Now, does something happen in another tetrad's portion of chromosomes number 1? Will break off and reattach with portion of the another chromosomes number 1. So the tetrads experience crossing over. Synapses crossing over only happen prophase 1 in meiosis. This does not occur in mitosis. So now, we're ready to move on into metaphase stage. On the left, metaphase of mitosis spindle fiber were actually pulled in these chromosomes randomly to the equator or middle line of the cell. And on the right side, spindle fiber were pulled tetrads randomly to the equator or middle line of the cell. And as we move on anaphase of mitosis, on the left side, spindle fiber were engaged in a tuggle war where they pulled the chromosomes, where the actual chromatids are pulled to the opposite end of the cell. And on the right-hand side, anaphase 1 of meiosis, the spindle fiber engaged on the tuggle war or actual pulled apart the tetrads. The tetrads separately with 1 chromosomes, and pulled to the left. 1 chromosomes of the tetrads pulled to the right. And so now we move on into the telophase of mitosis. In the left side, cytokinesis, the cytoplasm begin to split the cell in half. As that happen, the nucleus begin to regrow, and it's happen, the chromatids begin to unwind back to the loose linear version of DNA known as chromatin. And on the right side, telophase 1 of meiosis, cytokinesis begin to split the cell in half to create 2 cells. And sometimes the nucleus regrow, sometimes it doesn't. And if we stop evaluate mitosis cells on the left, this is the end result with cretentodiploid to diploid cells. Now, we're not finished with the cells in the right for meiosis. We're gonna continue that next. So the cell in the right is gonna continue through second round of meiosis. So now, here in prophase 2 of meiosis, now the nucleus simply dissolve. Now there's no more synapses, there's no more crossing over. So the next round of meiosis, prophase 2, metaphase 2, anaphase 2, and telophase 2, very similar to mitosis and what happened. Now, we're moving to the metaphase 2. Again, there was no second synapses, there was no second crossing over, so we're moving on to the metaphase 2 of meiosis, and the chromosomes are simply pulled on the middle line of equator of the cell by spindle fiber. And so what happened next as we move on anaphase number 2 of meiosis is that spindle fiber have a tuggle war pulled on the chromosome so hard that the chromatids are pulled opposite end of the cell. And as we move on telophase number 2 of meiosis, the cytoplasm begin to divide in a process known cytokinesis. But now, it's the 2 cells have been divided into 4 as the happening chromatids are unwinding back into the loose linear version of DNA known chromatin. And as that happening, the nucleus reforming in all 4 cells. So, notice what you have 4 cells cretin. Well, if we do a little end result, you notice not just 4 cells, just cretin, but the 4 cells are haploid. Now, let's look at the cells on the left. The cell on the left have the same amount of chromatin. As we started with, we put a note that 4 strands of DNA start, and notice there's 4 strands of DNA. And the 2 cells cretined by mitosis. Now, look the right and see. I know that there are 4 strands of DNA to start, but the new cells only have half the amount. They only have 2 strands of DNA. This is because of the second round of meiosis. We cretined haploid cells in the left. Notice, the cells cretined by mitosis are identical to false strand of black, 2 false strand of red of chromatin, but the cell on the right are genetically unique because of crossing over and the recombining of those portion of homologous chromosomes. Way back in prophase number 1, each cell is genetically unique. So as we wrap this up, the cells on the left retain by mitosis. They start diploid and they ended up diploid. But the cells undergo meiosis on the right, they started diploid, they ended up haploid. So that's all for today's video. Thank you for watching and I hope you learn about simulation of different stages of mitosis and meiosis.